So a little bit about Roche before I get into the details. Roche Sequencing is a business unit that got formed about 2 years ago in Roche Diagnostic business, and they invested more than $1 billion just the last year buying companies. And they're very serious to go and be successful in the sequencing. And for them, sequencing is not only measuring DNA sequence or analyze or build kits and chemistry and boxes and so. For Roche, it's all about human health. So it's all about delivering healthcare at the end, and that informatics and data part of it become extremely important because generation of the data will eventually become commoditized. There's a lot of competition now. There are many good companies out there that try to have the best chemistry, best technology. There are many interesting ones using nanopore or imaging or semiconductor-based DNA sequencing. And all of their goals is how to get the highest quality sequence and know what's happening in the DNA level. But eventually, what does it mean? Why does it matter is a very complex problem. Because in order to understand one genome, you need all the rest of genomes. You need to compare them. And you need to kind of continue looking at it. It's not just one-time processing. You need to look at the changes over time. One of the hottest applications in DNA sequencing that we are working on it, and several other companies also are investing heavily in it, is a concept that is called liquid biopsy. So if you are a cancer patient, God forbid, you know that it's very hard to monitor the cancer. Even the first diagnosis that you receive, it needs some very intrusive biopsy. For some cancers, it's extremely difficult, like lung cancer, the biopsy can be even life-threatening. And as the treatment starts, whether it is chemotherapy or whether it's immunotherapy, all sorts of therapies, they are interacting with the disease. And cancer, unfortunately, is a very smart disease, so it continues to mutate and evolve. And when you put it in an unfriendly environment with all those, chemicals and attack it, it evolves even faster. So cancer is not a static thing. It is, first of all, very heterogeneous. So you can have breast cancer, but it might be 10 different types of cancer based on their genetic profile. And it's also, each of them is evolving and mutating over time. So if you just do a physical, like local biopsy, first of all, you just see one time point and one location. So the picture you are getting is very imperfect, and you're causing a lot of cost and trouble and hurt to the patient, which might not be even possible. The concept of liquid biopsy is that you do a blood test. And the idea is that our plasma or blood goes everywhere in our bodies. And if there is a tumor somewhere, it's shedding its DNA in the bloodstream. If you also have some infection, it's probably some sample of that virus or bacteria in your bloodstream. So if we take a blood sample from a patient, if we have sensitive enough technology, we can figure out all these DNAs. And we can figure out, which cancer you have. And it's actually a much more fair representative of the cancer because it goes to all of your body. So you get actually a better sample representation of the types of mutation you have. And because it's not intrusive, you can actually do a blood test, liquid biopsy every month. There are cases that there are hypotheses that some cancers might exist in our body 10 years before we see any symptoms. So that test might help us to actually diagnose disease at stage 0 and then just prevent it instead of wait until it gets to stage 3 or 4 and becomes almost impossible to cure with all the complex treatments we have in the market. So liquid biopsy is one of the applications we're working on. I'm just giving it as one example. There are many others. Oncology, of course, is a big focus for Roche as well as other pharma companies. Infectious disease, rare genetic diseases, and reproductive health. How to make sure babies, are born healthy and help the parents to make right decisions. Those are some of the examples of applications that this technology we are building can really transform and improve healthcare. And also the transform I think, traditional diagnostic business model. Because when you think about healthcare, it's actually more sick care. People only go to doctors and they get engaged with the healthcare system when they're already sick, and it's usually too late in the process. With these advancements, healthcare will be much more integrated in our lives much earlier on, and consumers and people will get more responsible, and they get ownership of their own, managing their own health, and the cost of healthcare, instead of being spent all in the last 2 years in an unsuccessful way, will be spent in a way that you can actually create quality of life. So DNA sequencing is a complex process. In order to measure a DNA sequence and interpret it for an application, you need many components to work well together. Here is an extremely simplified picture to show you that there are at least these 4 components. There is a sample prep, sequencing, medical content application, and then analysis. So sample prep is all the steps from you get access to some specimen or biopsy, you want to purify the DNA out of it, then you get the DNA, then maybe you want to only look at these few genes that you care about, you capture those genes, you amplify them, you reduce the noise in the sample. These are all sample prep. After that, you basically put in a DNA sequencer, which are different technologies. They need some form of more kind of chemistry application to prepare the data further to enter to a DNA sequencer. DNA sequencing converts that chemistry to a bunch of binary data, zeros and ones. And those zeros and ones, are an incomplete picture of what is in your DNA. The example I usually give is that think about you have this beautiful image and then you shatter it to small pieces. These DNA sequencers, they give you those small pieces. You need to put them back together to construct that puzzle. And the reason that they cannot give you the whole puzzle is that it's just impossible to read all that 6 billion bases. And they have to chop it down to small pieces, read it at a time. They create a lot of redundancy to make sure they address all the noise and errors that still exist in these systems. So you end up receiving billions of small strings. Maybe each string is 100 characters. 100, we call it base. Base can be ACGT. It can be about 100, maybe 200, 300 bases, but this is the range. And then you have many billions of them. And when you put them back together, you get that full picture. And putting them back together is not a trivial problem either, because there's so much redundancy, there are errors, there's genetic variations in the population. So you need to take all of those into account. There are many heuristic and also probabilistic models of how to do it. But just this problem alone, sequence mapping, it can take days or weeks on a supercomputer for let's say tumor genome. In our platform, we have accelerated it to a few hours. We really optimized it in many levels, from how it is implemented, parallelized, the algorithms, and so on. So that is the first step. Okay, you get the sequence, and then the question is, okay, what does it mean in the medical application context that you are interested in? So the medical content are the kind of applications, or in the sequencing terminology, they call them assays. So for example, I care about these 14 genes because I know they carry some mutations that is actionable information, say, for a physician to guide a treatment. And that would be one application. So you narrow it down to that 14 mutations, for example, you care about, and then you look at all the literature about them, all the drugs that are available, the interaction between them, intersect it with how the patient is doing, and so on. So if you look at the market today, it started by the sequencing and sample prep. So there was so much excitement the last few years around, wow, it's the first time in the history we can actually do DNA sequencing, with few $100 in few hours. So still today, most of the excitement is there in terms of where people are buying things or building things. But starting to shift to more applications and data and informatics, because this problem is starting to get solved. And Roche, is basically not the first. It was the first mover. So first DNA sequencer in the market actually was Roche sequencer called 454. They had it even early 2000s, but they didn't I think give it enough attention. So it became less competitive to another company product in the market called Illumina. So Illumina became the leader in building those DNA sequencers. And Roche strategy now is that, okay, not only I want to beat Illumina here, they have a lot of investment and technologies here, but also I think this is going to be tiny compared to the real opportunity. Because the real opportunity is more in the data, informatics, and applications, which Roche is very poised to address, especially when you think about their presence in all the labs, hospitals, and working with the physicians. So if you look at the market size, the sequencing market size, DNA sequencing market size, that mostly is these two blocks, is about a $2 billion market today. But if you think about the diagnostic market that will get disrupted through the whole value chain, we are talking about, the estimate is between $20 to $40 billion market today. And this is not even including preventive healthcare and all the other things we discussed with the liquid biopsy. So Roche sequencing vision is what we call it a sample to answer vision. They are saying, today if you look at who buys DNA sequencers, they are very advanced, sophisticated labs. Think about, when IBM was in early days, who was buying computers, like those big mainframes. It was mostly very advanced labs who knew how to actually use these sophisticated computing technologies. Then what happened? Computers became cheaper, more accessible, personalized. And then what happened later? Internet came and all of them got connected. So similar trend is happening in DNA sequencing. So the initial DNA sequencers were costing millions of dollars, and only very big labs in the country could afford them. But now we are talking about sequencers that are about $50,000, and in a few years you're gonna have it, maybe a small device attached to your iPhone or smartphone that you will not even pay for it. And then how all of these genomes get aggregated and intersected and collected will become the main thing. So similar trend is happening, and Roche is investing in all of those to make it happen. They want to democratize the market and consumerize the technology. So this is just a snapshot of some of the main companies they acquired the last year to solve these different problems. We have several of them on the chemistry and reagent part. On the sequencing part, they acquired a company called Genia, which is also in Bay Area. It's a nanopore single cell sequencing. They also have a partnership with Pacific Biosciences. Both of them are trying to solve a bigger problem around length of DNA sequencing. So they are trying to improve how many sequences you can read at a time, from say 100 bases to about several thousands. And that will give us a much higher resolution of the DNA, especially when you think about complex events such as structure variations and how genome rearrangements happen. They can be very useful. On the medical applications, we are the winner now in the market in the NIPT, which is this non-invasive prenatal test. The mothers, after 6 weeks into pregnancy, they can do this blood test, and the same idea, some of the DNA of the fetus, the baby, is in the blood of mother. So by that DNA sequencing, we can actually diagnose some of the symptoms such as Down syndrome and others much earlier into pregnancy and inform the parent to make the right decision. We also have the liquid biopsy. We talked about it. We are also working with some very interesting companies, and acquired one of them, Signature, that their focus is actually building biobanks. So what they do is that they work with hospitals, and they want to create these longitudinal databases that can track the, say, course of cancer over time. They do all of these biopsies, they keep all the clinical data that is relevant to that. And that data is a very rich and unique data that can help us to figure out the patterns that can be used later, for drug discovery and so on. And of course, on the analytics is Bina, and the way this picture shows is as if it is a sequence of events, but the reality is that the analytics is kind of the infrastructure everything is plugging into. So we are building the informatics that keeps track of data through the workflow, and later the back end of aggregating, interpreting of that information. Any questions so far? Yeah, very quiet audience. Please.
What kind of systems are you using today to analyze, like Intel-based processor, RAM and code, and do you think it can go into much cheaper? Are you thinking of like GPUs or something like that?
Sure, I will talk about it. You have a question?
I keep it toward the end, mine is more quick.
No, go on. I want to make sure I answer the questions because if there's any part that is not.
So what is the diversity of products that Roche is offering today based on these technologies? I mean, these are acquisitions, but are a lot of these kind of consumable products that are being delivered to labs today, or are these so things that are still in research and pending FDA approval and things like that?
So let me answer these two questions before I forget them. We got to the rest. So one question was about what technology we use. We get to the product in the later slides to talk about more details, but today it's mostly based on just commodity computers. We provide the software environment both on an appliance, which is Intel processors. Basically, we work with Dell and we get their high-end servers. So we deploy the software there. We also have the software on Amazon Cloud for larger scale deployments. When we began the company, the first product we built was out of FPGA and GPUs. And the idea was, we started by very much focus on the compute aspect. We went through this evolution in the last 3-4 years. I think we evolved from compute to analysis and then what I want to call it intelligence. And what I mean by it is that we realized that there are so many problems to solve in the genomics informatics that we have to look kind of higher level. We couldn't just pick algorithms and accelerate them. That was not a winning commercial strategy. It could be still very impactful, but because the science of it is evolving rapidly, it is premature optimization maybe to look at custom hardware. So we instead needed a framework that is very flexible, adaptable, and kind of partner with the scientists to enable them to develop better tools. And then we figured out that we need to even help the whole organization to keep track of that data. So we built a lot of data management. We'll talk a little bit more about it. So the other question is what products Roche has in the market today. So these are nice bunch of acquisitions, but what are you guys selling? So some of them we are selling. These are actually Kappa Biosystems is the world leader in enzymes and the sample prep systems. Nimblegen is a very good market leader as well. Some of these are in the market, but these three are not. So these are some technologies that are acquired and being developed to be integrated with the rest of them. The PacBio and Genius sequencers, they are not out there. They will be. I cannot share timeline. The medical application, the NIPT is out there. Oncology will be out soon. Will be announced, I think, in the next couple of quarters. And Bina solution, part of it is out, and part of it we are building with the rest of the team. So the idea is for each of these components, they want to continue have a presence in the market and sell it as a standalone business to really learn from the market, make the product competitive with the other competitors in the market. But the bigger vision is that how all of them can get integrated into what we call it the dream machine. So the dream machine would be you put a drop of sample and then you get the insight out and everything is fully automated. Why the dream machine matters? Because that's how you democratize the market. So you can sell it to all the smaller labs and people who do not have the sophistication to put all of these together and make it work. So that's a longer term development, but basically this is the vision that the company has. You also have a question?
I was actually wondering exactly, so maybe you can give some examples about what analytics you can do, because my knowledge about this part is actually quite limited. The only thing I know maybe is the blast algorithm, things like that.
Yes, we'll get there. We'll talk a little bit more about that. Any questions? Yeah.
Can you explain a little bit about how you handle the amount of data? Because the amount of data is huge, and always when it comes to healthcare data, I think the security matters. So how you kind of handle kind of security of the data and the scale, the amount of data.
Sure.
Because you're focusing on data management and analytics.
Yep. We'll get to that.
Okay.
If I don't answer it, then let's talk about it. But I'll talk about it. So we can skip that. So the data challenge. So this is a picture that we created when we sat with one of our customers. It was a pharma company. We were looking at how they are consuming data and just the genomics data. And we realized it's just a very complex way they are consuming the data. They're doing some genomic experiments in-house. They're outsourcing some of it. They have collaborators with other companies in the field, and also just inside their company. Let's say they're working on several oncology drugs or other drugs, and they are producing data. And sometimes this data can be shared and leveraged, so they're also doing these collaborations. And it is a mess, really, because they cannot keep track of who is doing what and where the data sits. And these are very expensive experiments, doing a DNA sequencing and analyze and interpret it, and then not be able to really consume that data well is actually a big bottleneck, both in terms of money and time. And they were telling me, we are very secure now because we cannot find our data. Yes, security. So, the data is sitting in silos and flat file formats, and it is not even reproducible. Think about how horrible it is. We're talking about scientific experiment that should be used for drug discovery, and many of them are not even reproducible. So this is the big kind of data management issue we have. And talk about security, like the pharma companies are global, yeah. So they have a scientist sitting in China that has different security rules, working with a scientist in US. Many times data cannot even cross the border. So how do you solve all of these problems in an industry that is going more and more global, and the problems are so complex you cannot think about a small group of scientists can solve it. You need to bring different disciplines to work together. So we realized that the genomic data challenge has at least these 4 main pillars to it. It's not only about speed. I wish it was just speed, because that's where we started. It was easier to just solve that. So science first, that is like if you can improve the accuracy of information and quality of the data, that is huge. And people will pay for it, they will really appreciate it. The challenge is that this science is a work in progress, and a company cannot go and say, I'm going to solve the science problem. It's a whole community. So all the universities, the pharma companies, the academic organizations, they're all working on all the algorithms. There are thousands and thousands of algorithms to analyze the DNA data, and you need a framework to bring them together, maybe to compare them, maybe to guide the scientists which one to use, or how to compare their tool to the others out there. We do have a science team that actually are a very innovative team, but their goal is to keep us really at the cutting edge. Their goal is not to replace what our customers are also developing. So it's many times collaboration with the customers. The process is what we just talked about. So the data management, the governance, the processes of make sure the right person can see the right data at the right time and govern the data and manage it through the process. So this was one of the big achievements we had last year, that our system before was I would say, stateless system. The processing was very transactional. You come to this system, you say, okay, my input data, my algorithm, you get the output, done. Now the system is intelligent, so you come to it and you already have a project, and you can link some data to your project, and you already see a history of what happened already in that data, how you got it, what are the other analyses. You can go and reproduce them. Whatever you do will be registered in the system. And all this bookkeeping is very important, especially when you think about these applications like liquid biopsy, because it's no longer going to be transactional. You need intelligent systems that keep track of things, figure out the change, and can really help us do more complex analysis. So those are all parts of processes and data management. Technology, of course, is a very, very big area of focus for us. That's the big data aspect of it. So how to run these processes fast, especially when you think about point of care, like infectious disease, turnaround time is very important. throughput matters a lot, especially for research applications. Just last week of the year, one of our customers, pharma customers, they had this project. It was a purely research project. They looked at a database that was out there. It was a 20,000 exome data set. And they said, okay, we want to analyze it because we want to figure out if, they wanted to test the algorithm on it. It was a huge compute task, so we deployed it on 150 large compute nodes on Amazon, and we did the work with them in less than a week. So those are the things that are not trivial. You need a lot of experience with cloud computing and deploy these things easily and manage them securely. And the people is very important. Again, we are working on complex problems where many different types of people need to come together and solve them. The personas, let's say, that we work with. There is the IT people who care about the data needs to be governed and secured, and my compute resources should be used efficiently. Then there's the bioinformaticians who care about, I want to test my new algorithm and I have to analyze this data. Then you have the scientists and researchers who are not bioinformaticians, but they care about their biological question. They care about, okay, I want to know what is the role of this gene. So they need a completely different interface to this product, yeah? And then you have your, clinicians, people like a pathologist who just gets, this biopsy and then they want to see what is the report they have to send back to the physician. And then later maybe we can think about consumers. There are new companies that are thinking how to deliver this information to consumer. So it's like many different aspects of this product. It's the same data. You want to manage it, but interface it and give access to different functionalities on this data to different personas in the organization and enable the right collaboration between them. So that's also a very big component of our work.
So I'm curious about the what was the magnitude of the data size you mentioned in your last example?
I should look it up. Maybe after the talk I look it up. It's in my computer.
Sure, I was
and
just
share it
curious.
with you. Yeah. But when you think about the data size, one genome, so the sequence is not that big because you can think, okay, it's binary, ACGT, 2 bits is enough for each, so what's the big deal? The problem is that you also need to store all the scores with them. So when they do this DNA sequencing experiment, whether they use optics, imaging, semiconductor based, there's kind of a signal processing that happens there to tell you, it was A or C, and then there's these large quality scores that are real numbers attached to it. So when you combine that, suddenly the size of the file is about in order of a terabyte of information for one DNA sequence. So you can easily think, a few thousand people, you suddenly have petabytes of storage problem. Yeah. So one of our customers is AstraZeneca. One of their scientists, Johnson Johnson, he gave a very interesting talk last year, AGBT, and the video of the talk is actually available on our website if you're interested. So he talks about how they're using Bina to do drug discovery at AstraZeneca. It's a very large pharma company. They did a global deployment of our product in many different sites, and this is enabling that large collaboration. And he talks about how we accelerate the science. If I remember correctly, I think we reduced the time for whole genome from many days to a few hours for them. So that was like a big jump of performance in their projects. So he was saying, why do we use Bina? He mentioned these basically reasons. He said, bringing bioinformatics to the bench. Basically the scientists can also look at the data instead of always going back to this service center, which were the bioinformaticians. He said there are more than 16,000 ways to get things wrong, because when they put a workflow, there are so many different algorithms, each of them have so many different parameters, and it's just so many ways to get things wrong, and in order to make it reproducible, they wanted this system to keep track of things and optimize them. The scale and production, and we talked about expertise and skill availability and collaboration. So the way we work with our larger customers is that we look at it as a partnership, so we don't just give them the product and walk away. We sit with them, we design how they're going to use it. So it's similar to like how IBM actually work with big customers. So there's this consulting aspect to it, which is the expertise and skill availability. This type of skill set is rare, people who know how to analyze this data set. So the customers really appreciate having access to the team as well, which makes it difficult for us to scale very rapidly. So we need to work with fewer customers, but really deliver high quality service and product to them. So what do we do actually? So a little bit of terminology about the product. So someone makes a DNA sequencer, like Roche sequencer or Illumina sequencer, PacBio sequencers. There will be ways that you can create data. And thankfully, the field has converged to some standards that we have some file formats. For example, we don't care much what sequencer was used. We get this data in a format that we can consume in the later stage of analysis. What happens in the sequencer, we call it primary analysis, is mostly signal processing and creating that binary files that we talked about before. After that, you have secondary analysis and tertiary analysis. In secondary analysis, the goal is to construct that sequence back. So what is the full genome, for example, we saw. That needs a lot of computing, many algorithms. You need parallelization. You need to run it at scale. You need to keep track of all of the things we talked about. Then it gets even more interesting. You get to tertiary analysis, you want to actually interpret the data. For example, there is this concept of reference genome. Reference genome is supposed to be a healthy genome, So they put together this genome that represents what a healthy individual should look like. Then you sequence a new genome, you see, wow, there's some difference. So what those differences mean? Are they random? Are they benign? Are they disease-causing? If you're looking at a tumor, maybe you get a tumor sample and then a normal sample of that person. You want to compare and see, okay, what are the mutations that happen in tumor that caused it to be cancerous, And out of those mutations, do I care about all of them? Or maybe they're just passenger mutations and only one of them is a driver mutation that is causing all the troubles. So all of that discovery and interpretation is the tertiary analysis. In order to do that, you need to get access to a lot of scientific literature and content that are being produced on a daily basis in the field, All the PubMed literature, There are so many databases people are curating. And in your lab, you're also curating. Every experiment you do, every patient you deal with, you might learn something new. So you want to add to your knowledge base. So there's a big knowledge base component to this tertiary analysis. And there's also a speed problem because in order to basically query and ask these sophisticated questions, for example, a pharma company might want to look at all the clinical trials that are running and say, all the patients with this genotype and this type of disease, how did they respond to this drug? So you need to be able to traverse all these databases in a real time and get the relevant data with all the security we mentioned. So we did a lot of innovation there. We created our own unique indexing technology that creates a real-time experience for the user. So you can ask sophisticated questions that are, if you look at the logic, it's like hundreds of nested queries, and it's interactive. The user even does not realize, in a fraction of a second they get their answer, and that helps with this exploration and visualization analysis of the data. And eventually you want to give a clinical report back to the physician. So this is kind of a big picture of what we do. There was a question, okay, how do we manage this large data? So today we are deploying our product in two ways. One of them is on-premises. We call it like hybrid because we allow both models. On the on-premises model, we basically ship the servers and we put the software there, and we don't provide storage. So user usually have some high performance storage in-house, let's say Isilon disk and so on, and we integrate with their in-house storage. So with the data management layer sits on top of that, but we don't physically provide the storage. On the cloud side, at the moment we are expert in S3, Amazon storage. So the data usually just feeds there, and then we just analyze the data on S3 and write it back. So we have not solved the storage problem. We have been more focused on how to consume the data very quickly. So For example, even uploading the data to Amazon, how to make it in a way that is distributed, you can do it in a very efficient manner, how you can parallelize access to S3 and consume the data quickly and write it back. But can you compress the data further? I'm sure you can. We haven't spent that much time on it, but I think as this field goes forward, there are more intelligent ways of saving data, storing data, for example, look at differences, and all of that too. So the storage, I think, will probably not be a very big problem, but the idea of make these analyses real time and kind of interactive will continue to be a challenge as there will be more data.
Question?
Yeah.
How do you integrate with publicly available algorithms such as like for aligners and everything like that? And what have you guys done as far as benchmarking what your in-house developments created compared to standards in the field?
Right time for the question, because I had this slide exactly about that.
And I have another one.
Yeah.
Possible. So you are saying about reference genomes, like normal
Yeah.
human are many normal references, or is just ones that you know where to look for it, or you carry with you, or?
So the normal genome, we are not creating it. So there's these standard bodies like the NIST institutes and others. So it's a government effort, and a lot of research organizations are involved to put together the reference genome. There are few of them out there. They keep updating it, and the way they build it is that they look at several individuals. I think the first one was 13 individuals. And then they sequence them at depth, and they make sure the data is very accurate, and then assemble it. And then there's a database that goes with this reference genome, and that database called dbSNP includes all the variations that are frequent in the population. So there's this idea of single nucleotide polymorphism. So somewhere between 2 to 10 million places in our genome, we all have variations which are normal, like that maybe impacts our hair color or something, but it's not disease causing. So we have also a recording of that. So when you do the DNA sequencing, you map it against a reference genome, but also you look at all these databases to see, if it's just a frequent, if it's happening in less than 0.1% of the population, maybe I should look further, But if it's quite frequent, maybe I can ignore it. So there are all these adjacent databases that can help you to do the discovery.
And they don't store quality factors. I mean, they are kind of, this is what it is. You don't.
For the reference genome, I don't think there's quality yet. So let's talk about the science. So we talked about the overall engineering problem. But we also have a science team that actually develops algorithms and scientific data for the platform. And the reason for that was that we wanted to also innovate there, and we thought if we don't understand the science problem, how can we solve it for our customers? But we don't say that we're going to solve all the problems. It's just impossible. There's so many algorithms you can think of. But we have some very good innovations. First of all, we wanted to have a framework to compare algorithms, How do we know one algorithm is better than the other? And surprisingly, there is not an easy way to measure accuracy, because we even don't know the truth. Yeah? So you get a new DNA, you can compare it with that reference genome, but such a fuzzy, I think, comparison. And then all the ways to simulate the data, they're all so simplified. They are not really realistic. So we invested a lot of time on that. We created this algorithm called VARSIM, which was published last year in the Bioinformatics Journal, and I forgot how many times it has been downloaded. It has become very popular over the last year. And VARSIM is a framework to compare the aligners, variant colors, all of that. It has all this technology understanding, meaning that it asks you the Illumina sequencer data, how did you do sample prep, so and then it recreates the data in a realistic way to represent all the error modes and so on. And then it gets the truth, like it's all computationally basically calculated. And then it shows you how these different algorithms are performing compared to each other.
Does this work for RNA-seq too? And how about metagenomics?
RNA-seq, no. VARSIM is only the germline kind of analysis. For RNA and cancer, we have work in progress. I can we should exchange contacts. I'll let you know how it goes later. With metagenomics, we haven't spent that much time on it. It is on our to-do list this year because I think it's very interesting going forward. One of our highlights of our science team is the somatic mutations. So somatic mutations are the mutations that happens in tumor. They don't exist in our normal cells, but the reason that cancer happens is that something somewhere goes wrong. You get these mutations that disrupt the normal cell cycle growth and make it cancer. So those are somatic mutations, and we have built a machine learning approach to detect the somatic mutations in high accuracy. We participated in Dream Challenge, which was this global challenge of like who can detect somatic mutations in more accuracy, and Bina ranked first and second, and we won against so many big institutes such as Broad Institute, which is the world-leading institutes in bioinformatics, and just using our algorithm. So that was, I think, great encouragement for the team. So there are so many areas that if you are interested in statistics and computational biology, you can improve the status quo in bioinformatics. I think alignment, people spend a lot of time on alignment, but alignment is very technology specific. You need to really understand the chemistry and hardware very well. But when you get to the variant calling, I think it becomes more interesting, it becomes more application related. RNA-seq is very hot area because it's more functional than the genome. Genome is a silent state of what it is, but RNA is when it's expressed as a gene in action. So, and it's constantly changing. So think about the data deluge there. You might get your normal DNA sequence once in life, but your gene expression in every cell and every disease might happen, constantly. So if you're interested, we can talk more about it later. So we talked about the product, how it brings all of it together. And now being part of Roche, we have our own standalone product. We are selling it mostly to research organizations. Most of our customers are universities, pharma companies, and academic medical centers. But with Roche, the goal is to develop a clinical product that can be sold to all the labs and physicians and so on, and working closely with them. So few, food for thought. I was thinking, where value is going to get created in the informatics, genomics work. I thought about these three. I think these will be very big areas, big markets, big kind of opportunities to disrupt. One of them is just the infrastructure problem. I think we are working on it, but there are so many other opportunities to work on that for IBM and others. There is the content and insight. At the end, people want to know what this DNA, what this mutation means, and we have all these databases that are extremely noisy, extremely incomplete. I think it's just tip of the iceberg. So if there is an effort like Watson, for example, IBM Watson is trying to do it in a computational way. Many companies are investing even doing it manually, reading literature, curating data. That curation of literature and even real-world data is, I think, would be very valuable. And then the end-to-end integration. So how we can democratize the market, how we can make these technologies actually accessible and easy to use. So that was my slide. And I thought it would be 15 minutes, but it seems I thought 1 hour exactly.
Now I can ask the question.
So
the idea of liquid biopsy is very interesting for me. I'm an imaging expert, and we have a lot of imaging experts here. So my question is, we have been hearing, to be honest, about biomarkers that are going to basically change the way people look at cancer for a few years now. But still, if you go through most of the diseases, and I'm more toward the lupus prostate cancer, still there is no definitive biomarker, genomics biomarker that will tell us the stage of the disease. And I wanted to see if you can give us examples to basically in what areas we have definitive biomarkers now, which is basically the end result of all of this work, is that you want to create a definitive biomarker that would be used in a liquid biopsy or something. And what are the reasons of the, I don't know, maybe just from people on the outside, a little slower realization of this idea of just building a biomarker that can.
It's data. I think we still don't have enough data because for each cancer, there hasn't been enough experiments, enough clinical trials to pull things together and be able to find what is the biomarker. So when you think of biomarker, there are different ways to look at it. One of them is that pharma companies, they are more interested in the target. So they want to find a way that they can send that drug to your body or chemo in a way that only goes to those cancer cells. Because otherwise, they're attacking the whole body and it's even worse than cancer in many cases. So they look at targets that they can design an antibody or something that binds to. And they are finding some, like, all these excitements with immunotherapy now. Roche has few drugs in the clinical trial now. The idea is that they found that there's this, biomarkers or protein targets on the cell surface that they are fooling the immune system. So they are designing something to go bind there and dampen that effect, so the immune system now can actually detect cancer and kill it. So they're kind of, activating the immune system. So pharma look at it from drug delivery, Then on the science side, you go much deeper. You want actually to understand the biology of the cancer, because if you understand how actually it happened to begin with, you might be able to prevent it or do smarter intervention. And that you need to understand the driver mutation. That is the interesting thing, because when they do this DNA sequencing, they see many mutations happen in the cancer. But they don't know which one happened first, which one is the driver, and then how to correct it. So there are lots of, I think, scientific literature, but they haven't been translated to a drug now. The challenge is that if it is something very intercellular, how can you deliver something there? Most of the drugs still are big molecules, and it's hard to kind of interfere in a lower level. So I think they are investing a lot. All these pharma companies I see, they are doing large scale experiments. Roche Pharma actually. So it's very interesting. We're part of Roche Diagnostic, and then there's a big firewalls between the diagnostic and pharma. So Roche Pharma is as if it's another company. So now they are our customers actually. Roche Pharma and Genentech are Bina customers, and we had to go sell to them like another pharma. So they did actually two interesting acquisitions. One of them was just last week, Flatiron. So Flatiron is a company that got funded, I think, by Google Ventures earlier, and their idea is to create a database of real data evidence, basically, for cancer patients. And the other one is Foundation Medicine, that is capturing the genomic information of cancer. And Roche Pharma spent billions of dollars to just get access to this database, which is tiny. You're talking about thousands, maybe, I don't know, maybe less than 100,000 for sure. And this is how valuable this data is. So it shows to you that the access to data has been a big problem. When you think about how they design the clinical trials, they just look at a few thousand people,
So when you say we have a lot of the challenge of managing big data, we are typically in this context we are talking about the size of the data in one genome, but in terms of population, we don't have a lot.
We will,
We will.
I mean, we need to build it because we see it's coming,
Yeah.
I'm part of What's in Health, the new organization, and the question I have is with Bina. Do you have any experience already connecting to the public journals, the medical journals, and how are you right now dealing with that content? Is it static or downloading in terms of how you're updating it?
We are not doing any smart curation now. So we license a lot of data, we have all the major databases, we are licensing more, but we don't have a smart way of we screen the publication ourselves to curate. So this is an area we're actually extremely interested to see if there are ways to work together.
Yeah, I'm just curious. It looks like a lot of the work is to help with the early detection side of things, looking at the genomic level. I'm just wondering if you integrate different data sources, a lot of heterogeneous data. Are you looking at image features, different imaging data? Are you guys looking at radiogenomics field as well?
Yeah. So it is coming, yeah. So at Roche, of course, they care a lot about it. They have a large tissue diagnostic business, staining, all of the imaging. They have a large molecular diagnostic business with PCR, other assays, and it will eventually be all integrated. So when I think about that dream machine, I think it's more than DNA sequencing. It's going to integrate all of that. But it takes time. First, we need to integrate in this level. Each of them also have their own advancement, but there are some consolidations in a higher level on the strategic level that we are talking about how all of them come together.
Yeah, I'm still really curious about the CAPSIC, the blood test for cancer.
Yeah.
It's been sort of shown in lung cancer. I'm not sure the accuracy. What other cancers? As in looking at other cancers right now to see that blood test will work for detection.
CAPSIC is a liquid biopsy technology,
Correct.
so we just got the licensed technology from Stanford,
Yeah.
and we are working on it now. So it will be launched as a product for some particular cancers. Yeah. I cannot share more, but I think you'll know some. Any other questions? Yeah.
Question on application, like for oncology you already covered. I'm wondering about detecting what's causing allergies and possibly finding a cure for allergies and asthma using this technology.
I'm not the best person, I think, to answer that. Sorry, but I can follow up with you later. Can ask you for it.
So thank you very much, Narges, for coming and giving us the lay of the land in field of work. This is
Thank you.
from
Very
IBM,
nice.
and we thank you for your